All right, good morning, everybody. So today I want to go ahead and go over cellular transportation. There's a big chunk of this module on it. So I want to go over some different parts of it. All right, so let's go ahead and start. The cellular transportation, the bulk of it's going to take place in the cell membrane. So cell membrane, all cells have a cell membrane. It's the very outside of the cell. It's what we call selectively permeable. So some things can go in, some things can go out, but it can kind of prevent, it restricts what can and cannot go in there. If you remember from the first module, we talked about lipids. So part of lipids are these phospholipids. That's what makes up the cell membrane. So you have a hydrophobic end and a hydrophilic end of these phospholipids. And if you throw them all together in a bucket, they're just naturally going to form like this, which is basically a cross section of the cell membrane. All right, and here's kind of a 3D view of that looks like. So again, every single cell has this. And then on top of this, there's going to be almost like jelly bean-like structures embedded in, and some other things, proteins, protein channels, all sorts of other stuff. But this is the bulk of the cell membrane. All right, so there's two main types of transportation. There's what we call passive transportation, and then there's active transportation. So there's kind of two subsets of passive transportation. We'll talk about that in a second. But so first of all, passive. So think about it, passive. If you're passive, you're not using any energy. Okay, this is just going to occur. So passive transportation is the movement of substances from high concentration to low concentration. So it's going to go with the concentration gradient. That's just a fancy way of saying it's going with the natural flow of things. So think of it that. So if you take like food dye or something and just put a drop of it in water, it's just going to flow from the high concentration of the drop, and then it's going to spread out to the low concentration everywhere else. Think about it that way. So simple diffusion, that's what I just mentioned, high to low concentration. It's going to go right through this lipid bilayer. So whatever particle this is, whether it's water or something else, that it's going to take in high concentration here, low concentration here. It's going to move from high to low. Really simple. Okay. Then there's what we call facilitated diffusion. So facilitated, think of that word facilitated as like assisted diffusion. So this is for larger particles that can't fit through the cell, the phospholipid bilayer. So instead, they have to use these protein channels. Okay. And basically this allows for larger particles to go through, like I said, that can't fit through. So same thing, high to low concentration, but instead you're going through these channels here that are proteins. Okay, and there's another type we're going to go into more detail after, which is osmosis. It's just specifically for water. It's just passive transportation of water. I'll talk about that in a minute. All right, and then the last one is active transportation. So active transportation, okay, is going to require energy. Now, energy in a cell is what we call ATP. Okay, and then it's also going to use a protein. So facilitated diffusion does use a protein, but it's still passive, doesn't require energy. Active transportation, always going to use a protein. It's always going to use energy. And instead of going from high to low concentration, it goes from low to high concentration. And instead of moving with the concentration gradient, with the natural flow, it moves against the concentration gradient or against the natural flow. So a lot of times on a test, you'll see a picture like this, and it might be labeled, W, X, Y, Z, and, which one is active transportation. So, just something to keep in mind. So here you can see low concentration, it's moving, the arrow's pointing to the high concentration. Okay, what also gives it away is the energy there. This could say ATP. Okay, just keep that in mind. All right, and that was the last one. Okay, so I'm going to go ahead and move on to osmosis. Okay, so osmosis is a passive transportation. We just talked about passive transportation. So it goes with the concentration gradient, goes from high to low concentration. It does not require energy. Okay, this is just specific to water. So it's diffusion of water, movement of water from high to low concentration. Okay, and when we're talking about osmosis, there's three different types of solutions that can occur. There's what we call hypertonic, hypotonic, and then isotonic. So when a cell is in a hypertonic solution, okay, this means there's more solute in the solution than the cell. Okay, what that means is you have a cell in a solution of, it's going to be salt water or sugar water or something else. There's more stuff, there's more dissolved things in the solution, in the liquid, than there is in the cell. So because of that, everything, like I said, everything's going to want to even out. You're going to want to move from high concentration to low concentration. So the higher concentration of water is going to be in the cells. So that water is going to want to move out into the solution, okay? Because there's less water, there's more stuff in the solution. So it's going to move high to low. So that water moving out, it's going to shrink. Okay, those cells are going to shrink. Okay, so kind of give you like a real world example of this. If sometimes if you get like a mouth sore or something, or you had some dental work and you might have some swelling in your mouth, they often tell you to kind of swish salt water, or if you have a sore throat, or gurgle salt water. So when you do that, when you're gurgling salt water or swishing salt water in your mouth, it's going to pull the liquid out of those swollen areas into that salt water, and that helps reduce the swelling and help alleviate things. Okay, this is also the reason, especially more so up north when there's a lot of snow and they put salt on the roads. So that salt is going to end up killing the plants because it dehydrates the plants, pulls the water out of the plants, okay, and they can't survive. A hypotonic solution is the exact opposite. Okay, so this is be like, pure water, distilled water, something like that. There's nothing dissolved in the solution. Okay, it's just pure water. So that means the cells are going to have more stuff in it. Okay, so the water is going to move into the cell and cause the cell to expand. Now, in animal cells, if this happens too much, the cells can burst. Okay, so this in a plant cell, plant cells have a little extra protection. Obviously, they need water. They need to hold on to that extra water. So they have a cell wall. So water moves in. It doesn't burst. It just swells. Okay, so to kind of give you a real world example, if you're ever, really sick or whatever, and you go to the hospital and you need an IV for some fluids, okay, the solution they give you that goes directly in your bloodstream is not pure water. Okay, if they put pure water in you, that would be bad. Your blood cells would fill up and they could explode. They put a saline solution in you. So the saline solution has just like a little bit of salt in there. And the whole point of that is to match the solute content that's in your blood cells. So now the blood cells are in the same solute content as the solution that the blood cells are in. So it effectively hydrates you instead of, hurting your cells. So that kind of brings us to our next solution when it's equal. So when you have equal solute in, equal solute out, that's what we call an isotonic solution. Okay, so water does move in and water moves out. It's just at the same rate. Okay, and then just some other things I wanted to touch on. So, this kind of all has to do with surface area, just in general. So the reason why cells are so small is because they don't have a large enough surface area. If you had a really large cell, that cell wouldn't be able to take in enough water and get rid of waste efficiently enough because it wouldn't have a large enough surface area. So just think of it this way, like as far as your EOC goes, the larger surface area, the more efficient a cell is, not just a cell, but different like organelles, which is the reason why some organelles have kind of like a folded structure that you'll see when we get into the different types of cells. All right, and that's pretty much everything. All right, I'm going to go ahead and stop this. If you got any questions, let me know. Otherwise, you have a good day.